GOBii is the Genomic Open Source Breeding Informatics Initiative. Everything is in the name. There's a lot in the name. So we're all about breeding. We're focused on breeders and what breeders need. Everything we do is open source, so you're welcome to take it home at the end of this week and try it out. Warning, it's still difficult to set up and use without a lot of training or assistance from us, but you're very welcome to do that. And we're all about managing genomics data, but not just genomics data, also any kind of genotyping data, any scale of genotype. It's a 5-year project. It ends in 2020. We have extension for 2 years through the Excellence in Breeding program beyond that. But we are looking for new projects, new collaborations, new ways of working with people. So keep that in mind. So why was this project funded? This was a Bill and Melinda Gates analysis of yield from 1961 to 2007, showing yields for major crops in USA, China, India, and Africa. And while USA and China have kind of gone through the roof there, India, not so much impressive yield increases, and Africa was practically negligible yield increase. Now, Kelly talked a bit before about why that is. I mean, it's because they're using old varieties, but the hope is that by using genomic selection, it can be one of the tools in the breeder's toolbox to be able to start increasing those yields. But you can't do genomic selection without good data management. So that's where we come in. Kelly already gave our mission. It's to transform breeding using genomic marker-assisted selection as part of routine breeding programs. And again, routine does not just mean having a single experiment. What we're trying to do here is put together all of that complex logistics that needs to be able to happen to be able to implement genomic selection within a breeding cycle. So this is the components that we believe we need to focus on as part of this project. So we're here, we're doing the genotyping data management, and we're also doing some of the high throughput analysis tools and visualization tools. The foundation of this is to have a good breeding management system. We need high throughput sampling and shipping systems, a good genotyping platform, and most of all, we need these APIs that Kelly was talking about to hold everything together. Now, if you're doing math, you can get away with Excel spreadsheets, but really when you move to genomic selection implementation, you need all of these tightly tied together and running as a well-oiled machine. So this is our community. It's one of our advantages. We're not just based here at BTI and Cornell. We have GOBii employees at CIMMYT who work on maize and wheat, at IRI who work on rice, and ICRISAT who work on sorghum and chickpea. Then we also collaborate with a company called Diversity Arrays Technology in Australia and the James Hutton Institute in Scotland on tool development. So they've really helped us with analysis and visualization tools. And we fit here. Maybe it wasn't obvious from Kelly's talk, but we're not actually funded by Module 5 of Excellence in Breeding, but we're under the umbrella with that same, and in future we will get funding through Excellence in Breeding. Several databases out there that can manage genotyping data. You've seen Cassava Base had some genotyping data capabilities. There's Dataverse, Germinate's another good one. GDMS is within the BMS database. But they all tend to handle lower volumes of genotyping data and not the huge scale of genomics data that we can handle. And we can handle not just the huge scales of genomics data, but also your, one or two samples by 10 SSR markers as well. And we can handle any genotyping technology, whether it's sequencing, SSR data, something run on a gel, we can manage that data within our system. And we can also, we're not just talking about depositing data into a data set and extracting it. You can search for a sample or for a marker across all of your data in the genotyping system. So I'm going to talk a bit about technologies, just to say, we started out this project just testing different open source databases. This shows 4 different technologies: MonadDB, HDF5, MariaDB, Postgres, and the time that it takes to load a data set. And this has been updated since this time. We have a few other technologies we've tested, but HDF5 time and time again comes out as the best database methodology for being able to store genotyping data. So for our standard data set that we were testing, 5,000 samples, 32 million markers, it can load in under an hour, which might seem like a long time, but you compare it to the other systems, and it's really the fastest one out there. And then this just shows the extract. This is time in minutes, and then data sets of increasing size across the bottom there. MonADB just stopped working at a certain point. Postgres actually did pretty well in this scenario, but HDF5 in every scenario we've looked at is the fastest way you can extract your data as well. Basically, we're designed for extract performance in mind. Kelly showed part of this. What I'm going to emphasize here is the client interacts with the database system via two different UIs, which you will see this afternoon. Dataloader, and you can load in multiple different formats: IUPAC, VCF, dominant, codominant, SSR alleles in text, CSV, or HapMap files. The data goes into the system, gets broken up. The metadata goes into Postgres. The big genotyping files go into HDF5. And they're put into two different orientations so that you can extract in two different orientations. So whatever orientation you want to extract in, it'll be really fast. And then for the extract, we have two formats right now. We'll probably be adding more, but these are Flapjack for the downstream analysis tools we have, and HapMap. So now I'm going to get into more of how the system's set up, how you would organize the data in your system. So the data is organized by project, and by project we mean a genotyping project, not a field project, which consists of a set of samples. Those samples can be run on one technology or more than one technology. You might want to run both Illumina and CASP, say, on one set of samples. So you store all of those under your experiment. And then, especially for GBS, you can have multiple data sets. You might have a raw data set, you might clean it, you might do an imputation. And so all of the different data sets can also be stored and organized underneath those experiments. Now, we do have some germplasm tables. They're not extensive. We're assuming that most people will have their own breeding management systems and would use some kind of external ID that would link together GOBii with the breeding management system. But if you don't have that information, or if there's information you want to store in here to help breeders do an extraction, then we have some germplasm metadata you can store here. And the markers are associated with platforms, and so if you have the same marker name in two different platforms, it will get a unique ID. So you can sort out markers that have similar names across platforms, same names across platforms. You can underlie, you can give your marker a variant ID. So this is if 10 different markers all interrogate the same SNP, then you can give them the same variant ID. So they're all grouped together. You can map to genetic and physical maps, and you can group your markers into marker groups. And this is really useful. If you've got a QTL with 5 markers underlying it, you can group those together as one marker group, extract by that one marker group, and use that in Flapjack analysis as a QTL. So back to the concept of sample, we actually struggled with this a little bit to begin with. What was a sample? Was it the sample that was taken from the plant, or was it a sample taken from an individual plant in a bulk? And what we ended up with is landing on the sample being whatever went into the plate that went to the vendor. This simplifies it a little bit. So we're ignoring everything that's happening out here, but there's going to be a sample in a plate which has a well and position, or a sample in a tube that has a number. Those two pieces of information gives uniqueness of that sample within the project. And then we also ignore all of the downstream dilutions that happens in the lab, and we just assume the vendor LIMS system handles all of that. And just a little bit more about what germplasm names we store. Most important here is the sample name, because the projects consist of the sample. We do have this concept of a DNA run name. So the sample name will be sent to the vendor. Sometimes the vendor will actually send back a different name. If you've had that experience, they'll append some digit on top of it, or add the well name and well position. And so we can accommodate that name that's coming from the vendor and link it back to the data. we have this concept of an external code, and this is the external code comes from the breeding management system. So this is a really important piece of information. This is what's going to connect two different systems together. Most of our users are using the plot ID in this space, so the plot ID from which the sample was taken for this one. And then the germplasm name, just a common name that can be used to extract data. Okay, I'm going to skip pretty fast through these because Deb's going to show you how that looks in real life this afternoon. But this shows the GOBii data loader. This is where you set up your project, an experiment and data set. You can set up map sets, and they can be genetic or physical map sets. So those are just maps, genetic and physical maps. You can define your marker groups in here. So you see that you've got a marker group name, a disease resistance, and then you just load markers to it and the favorable alleles. Then you have this section here which basically sets up all of your support information that you need for a project. So what are my standard platforms? And we preload GOBii with these, but you can always add your own. So standard platforms, CASP, Illumina, sequencing, GBS, things like that. Controlled vocabularies for germplasm, so species are all controlled in here. Germplasm generation, so F2, BC1, and all of your contacts. And then here, this is the magical place where you get to load your data. So this just shows how you would load a matrix into GOBii. You've defined already the projects, the experiments, and the data sets for this. And then you preview the file that you're being loaded. You can preview it from your desktop, or you can put a path in for a file for a server. And then you just define where your markers are, where your samples are, and then where your first data point is. And then beyond then, GOBii can load the data to the database.